Hello students, welcome to EPG Pathsala. My name is Anant Narayan Bhatt and I work as a scientist in DRDO lab, Institute of Nuclear Medicine and Allied Sciences in Delhi. Today we are going to talk about enzyme cooperativity from paper Molecular Enzymology and Protein Engineering in Biophysics. So in this chapter, we will learn about following aspects of enzyme cooperativity. The first is Hill's coefficient, which is derived from the Hill's equation, and then cooperativity, and then the types of cooperativity, like the positive cooperativity or the negative cooperativity. Then how these cooperativity, I mean, especially in terms of binding of oxygen to hemoglobin molecule when takes place, it is defined in terms of models. So we will be talking about the concerted model and the sequential model of enzyme cooperativity or positive cooperativity. So when we talk about enzyme cooperativity, it is basically a phenomena in which the shape of one subunit of an enzyme consisting of several subunits, I mean in multimeric proteins, allosteric enzymes, when the substrate bind is altered by the binding of the ligand. So this ligand can be either the substrate itself or some other molecule which alters the substrate binding on the enzyme. So like in case of receptor, it can be ligand, or in case of enzymes, it can be the substrate molecule or the effector molecule or the inhibitor molecules. So if the ligand induced change in shape facilitates the binding of substrate to same or other subunit, substrate to the same subunit or other subunit, the effect is called positive cooperativity. However, in negative cooperativity, the binding of a molecule to the first subunit inhibits the binding of substrate to either to the same subunit or again to the other subunit. So this is called negative cooperativity. So therefore, in multi-subunit enzymes, binding of substrate is cooperative and it shows sigmoidal curve. So this sigmoidal curve is a characteristic of cooperative binding or cooperative enzyme kinetics. So this sigmoidal curve, unlike the so these allosteric enzymes, they show sigmoidal curve unlike the hyperbolic curve which is obtained in Michaelis-Menten enzymes. So to start with, when we talk about Hill's coefficient, the enzyme with single active site, they follows the Michaelis-Menten model. However, Michaelis-Menten model cannot define the enzyme kinetics of the enzymes which have multiple active sites, like we talked about the enzymes which have multiple subunits like hemoglobin, or the allosterically regulated enzymes. So it cannot, Michaelis-Menten model cannot define the kinetics of such enzymes. So allosteric enzymes, they usually display a sigmoidal curve, unlike the hyperbolic curve which we talked about in case of Michaelis-Menten model. So when the curve is plotted between the reaction velocity Vi and the substrate concentration, then it, as compared to hyperbolic, it results into a sigmoidal curve, and that is called the cooperativity. And when this is simplified or made linear, then it gives the Hill's equation. So here in this graph, you can see that there is a comparative graph which is plotted between reaction velocity and substrate concentration. The enzymes which follows Michaelis-Menten model, you can see that they are showing hyperbolic curve, which is shown in black color. However, the enzyme which has multiple sites and then it can bind multiple substrates, it generally shows a sigmoidal curve, which is shown in red color. So you can see very easily that how the enzymes which follow Michaelis-Menten model, which can bind only one substrate, and the enzymes which follows enzyme cooperativity shows sigmoidal curve and can bind multiple substrates. The best example of enzyme cooperativity where multiple molecules can be bound to one enzyme molecule is the tetrameric hemoglobin, where the oxygen binding curve is the best studied model as a sigmoidal curve. Here the 4 molecule of oxygen can easily bind one by one to different sites, and that shows a perfect sigmoidal curve, and that is the best model for studying the enzyme cooperativity. As we discussed before this, that Michaelis-Menten enzymes, Michaelis-Menten model cannot be fitted to the enzymes which has multiple binding sites or the allosteric proteins and which show a cooperative binding curve. So the Michaelis-Menten model is not a suited model for those enzymes. Therefore, the A.V. Hill gave an equation, and the graphical representation, it is basically the equation derived from the graphical representation, which is a sigmoidal curve. So he gave this model basically to understand the cooperative binding of oxygen to hemoglobin. So in this equation, which is log V1 oblique Vmax minus V1 is equal to n log substrate concentration minus log K dash, this equation states that when substrate concentration is low relative to K dash, the initial reaction velocity increases as the nth power of substrate concentration. So here the K dash is a complex constant. Using this equation, a graph was plotted between the log V1 oblique Vmax minus V1 and log substrate concentration, which basically linearizes the sigmoidal curve. So when we do the graphical representation of the Hill's equation, we find that and when we plot the graph between log V1 oblique Vmax minus V1 and the substrate log substrate concentration, we find a linear graph, and this which is used to calculate the substrate concentration at which enzyme attain half maximum velocity, and the degree of cooperativity of Hill's coefficient, which is basically the n, the number n, and the slope, a perpendicular line which is dropped from at the velocity where the velocity is 0 is actually the s, which is called a value called S50. So basically the slope of this linear line, which is n, is basically called Hill's coefficient. When we do the graphical representation of Hill's equation and find out the slope of that linear line, we can find out the Hill's coefficient value. And a perpendicular line dropped on x-axis from the point where y-axis log V1 oblique Vmax minus V1 is 0 intersects at a substrate concentration termed S50. So this substrate concentration S50 is also generally calculated for the hemoglobin and oxygen binding. There it is called pressure 50. I mean the oxygen pressure, 50% oxygen pressure at which the saturation of hemoglobin is basically calculated. So in terms of hemoglobin, it is called P50. But here when we are studying the binding of any other allosteric enzyme with substrate, then this value will be called S50. So in this graph, the V1 represents the initial velocity, Vmax represents the maximum velocity, S represents the substrate concentration, K dash is basically the complex constant, and N, as we discussed, it is a Hill's coefficient. So in Hill's equation, as we discussed about, which is for cooperative enzymes, it is basically the graphical representation is used to calculate the degree of cooperativity in allosteric enzymes and the value of Hill's coefficient in enzymatic reactions. So Hill equation is basically give you an idea about that what is the number of substrate which is binding to the allosteric enzymes, how many multiple active sites it may have, and then what is the cooperativity, how basically it is facilitating the binding of one substrate, how it is facilitating the binding of other substrates. So Hill coefficient, which comes out of the Hill's equation, is an empirical parameter whose value is a number of the whose value is basically a function of the number, kind and strength of the interactions of the multiple substrate binding sites on the enzyme. So overall, Hill's equation basically gives an idea about that how many number of molecules are binding, and then this binding of first molecule on the active site, how it is facilitating the binding of other molecules on the other active sites, and what is the cooperativity between binding and then the enzyme kinetics. As we discussed above that Hill's coefficient, n is basically the most important part of the Hill's equation, which tells us about the nature of the enzyme. So when the value of the nature of the enzyme depends on the value of n. So when the value of n is equal to 1, that means the reaction is not showing any cooperativity and all binding sites behave independently. So the one substrate binding to one site, it is not going to affect the binding of substrate on the other side. However, if n is equal to more than 1, so that means the enzyme is showing positive cooperativity, and where the binding of the first substrate molecule enhances the affinity of the enzyme for binding other substrate molecules. So this is called basically positive cooperativity, and in this scenario the curve will be very perfectly sigmoidal. So the greater the value of n, the higher will be the degree of cooperativity, and there will be more sigmoidal curve which will be plotted between the reaction velocity as well as the substrate concentration. So however, if the value of n is less than 1, that means the reaction is not at all showing any cooperativity. And what does it mean? That the binding of substrate at one active site is not at all going to facilitate the binding of other substrate on other substrate molecules. So this is how very nicely designed or evolved during evolution in allosteric enzymes, that the binding of one substrate molecule can really facilitate the binding of other substrate molecules, and that is how the allosteric enzymes are playing a very important or major role in regulating several biochemical reactions in the cells. So this kinetic behavior, which is basically, we can see like the binding of one substrate molecule is facilitating the binding of other substrate molecule, is this kinetic change, basically this reaction kinetics occurs because of structural change in the molecules. So in the absence of substrate, the enzyme exists almost entirely in T state, which is called tense state. However, the binding of substrate molecule to the enzyme, the first molecule, binding of first substrate molecule to the enzyme converts it from the T state to the R state. So a transition from T to R favored by substrate binding to one site, and this will increase the enzyme activity of the remaining other sites at which the substrate has to come and bind. So this will lead to an overall increase in enzyme activity. So this important property is basically called enzyme cooperativity, and because of this subunit, all the subunits, they cooperate the binding of the substrate to the enzyme molecules in such type of enzymes. And then an increase in substrate concentration, it always favors the transition from T state to R state. So in multimeric proteins or enzymes, the binding of a ligand or substrate in case of enzymes to one site or subunit may alter the binding of the same or other molecule on other site or subunit. So this phenomena in multimeric proteins is called cooperativity. So when the reaction velocity is plotted against the substrate concentration, so with increase in substrate concentration, the binding of the substrate molecule on the enzyme or the binding of the ligand on the receptor is facilitated. It increases, and both these parameters, they show a sigmoidal curve when they are plotted against each other. So cooperativity can be seen in both enzymes and receptors having multiple substrate or ligand binding sites. That is the one thing. And then the concept of cooperativity is applicable only in the case of molecules having more than one substrate or ligand binding sites. So based on types of substrate or ligand, cooperativity can be of two types. So enzyme cooperativity can be of two types based on that what kind of substrates are binding on the enzyme. So if a ligand, like if a substrate molecule for the enzyme or ligand for the receptor, when it influences the binding of similar kind of other molecules also, like oxygen in case of hemoglobin. So when oxygen binds to hemoglobin, it facilitates the binding of other oxygen molecules. So this kind of cooperativity is called basically homotropic cooperativity, because binding of the oxygen molecule is facilitating the binding of other oxygen molecules to the active site of enzyme. So here in the figure we can see this is the multimeric protein, which is probably in tense state, and then when the first molecule of oxygen binds to the tense state, then the partially saturated tense state basically brings a structural change in the entire tetrameric enzyme, and then it brings it to completely R state, where all the oxygen molecules or all the similar substrates can bind on different subunits. So basically homotropic cooperativity can be said it is the cooperativity in terms of substrate, when the one molecule of substrate A facilitates the binding of other molecules of substrate A on the active sites of enzyme, then it's called homotropic cooperativity. So the other type of cooperativity based on the substrates which binds to the enzyme molecule is called heterotropic cooperativity. So in heterotropic cooperativity, when substrate A facilitates the binding of substrate B on the other subunits, then it's called heterotropic cooperativity. It can be substrate or it can be ligands in case of receptors. So in figure you can see when the substrate 1 is binding on the active site of one subunit, what it does, like it brings a structural change in other subunits, and then other subunits can easily bind to substrate 2. So in this case you can see like there are two different substrates, and the binding of substrate 1 is facilitating the binding of substrate 2. So there is something inverse to cooperativity that is called non-cooperativity. So if the ligand binding to any one site does not affect the other site in multimeric enzymes or receptor, then it is called non-cooperative. So the cooperativity is basically in allosteric enzymes, it can be of two different types. So positive cooperativity and negative cooperativity. So to start with positive cooperativity, when the binding of a substrate or ligand molecule facilitates the binding of same or other substrate or ligand molecule, then it is called positive cooperativity. So the value of Hill's coefficient in this condition in positive cooperativity is always more than 1. Then the best example of positive cooperativity is the binding of oxygen to hemoglobin molecule. So when there is increase in the concentration of oxygen, when there is increase in the concentration of oxygen, it facilitates, or pressure of the oxygen, it facilitates the binding of other oxygen molecules to hemoglobin. And then Christian Bohr, who have studied this positive cooperativity of oxygen binding to hemoglobin significantly, he showed that the carbon dioxide molecule, when it binds to the hemoglobin molecule, it inhibits the binding of oxygen molecule to hemoglobin. He also noticed that this phenomena, together with the observation that hemoglobin's affinity for oxygen increases with increase in pH, and this phenomena is known as Bohr effect. However, the binding of carbon dioxide, which inhibits the binding of oxygen to hemoglobin, that acts as a negative effector molecule, or effector molecule in case of allosteric regulation. So Christian Bohr, who studied the binding of oxygen to hemoglobin in detail, when he started studying the binding of hemoglobin to oxygen under different physiological conditions, what he found, and when he plotted the graph, what he found, that hemoglobin saturation with oxygen as a function of partial pressure of oxygen. So he obtained a sigmoidal curve between oxygen pressure as well as the relative velocity of the reaction of oxygen binding to hemoglobin. So what this indicates, he found a sigmoidal curve, and this sigmoidal curve, what does it indicate? That the oxygen binding to hemoglobin, it facilitates the oxygen binding to hemoglobin facilitates the binding of more oxygen to the hemoglobin. So this graph until all binding sites are saturated, this binding is continuously going on. So if you do careful analysis of the binding curve of hemoglobin, it indicates that the binding of oxygen is basically dependent on binding of oxygen to hemoglobin is basically dependent on the oxygen concentration. So the affinity of oxygen is low at low concentration. You can see in the first phase of sigmoidal curve that the oxygen pressure is low, so the binding of oxygen to hemoglobin is also low. In the transition phase, you can see that it is going very high at certain concentration, that can be P50 or as we discussed in case of linearization of Hill's equation, that at P50 concentration probably the binding of oxygen to hemoglobin is very steadily increasing, and then it gets completely saturated. So you obtain at high oxygen pressure, most of the hemoglobin active site is completely occupied by the oxygen. So as we discussed the oxygen binding to the hemoglobin, and we discussed also that the oxygen binding to hemoglobin is one of the most studied model, one of the most studied enzyme cooperativity phenomena. So there are various models which have been proposed for the binding of oxygen to hemoglobin. How basically the change from the tense state to the relaxed state occurs, and then how oxygen molecule binds to different state to different subunits, and then it changes the T conformation to R conformation. So majority of the models which are given, they basically talk about the conversion from T state to R state only. So the first model which is given for this is the concerted model, and this model is known as symmetry model also, or MWC model also. So this model basically it emphasizes that enzyme subunits are connected in such a way that a conformational change in one subunit is necessarily conferred to all other subunits. So this model tells that the subunits must exist in the same conformation. For example, in hemoglobin, the tetramer changes conformation together to the R state. After 4 oxygen molecules, when it binds to all 4 monomers, the transition from the T state occurs, transition from T state brings it into the R state, and this occurs in one step only. So at each label, the binding of oxygen binding of oxygen molecule, there is equilibrium between the T state and R state also, but what this model proposes, this equilibrium shifts from strongly favoring T state, no oxygen bound state, which is, to strongly favoring R state, which is fully oxygen loaded, in one go. So overall oxygen binding shifts the equilibrium towards the R state, which is like the general understanding in all the models, and this means that a high oxygen levels. So this model also suggests that at high oxygen concentration, the R form of the hemoglobin will be prevalent. However, at low oxygen concentration, it is predominantly T form which will be prevalent. The next model of oxygen binding to hemoglobin, which explains the oxygen binding to hemoglobin, is sequential model of oxygen binding on the surface of hemoglobin. So in this model, what basically it tells, which is very different from the previous model, is that here it is not necessary that all the monomers in any tetrameric enzyme may have the same conformation. So oxygen binds to one subunit, and then the binding of oxygen to the one subunit will bring the structural change only in that monomeric subunit only. It is not going to bring the change in other subunits. So in this model, it is not necessary that all enzyme subunit they will have similar conformation in multi-subunit structure, and substrate bind through an induced fit. So when it binds, basically changes the structure. So like in sequential model, for example, in hemoglobin, the 4 monomers, they change the conformation from R state to change the conformation from T state to R state, and this change occurs one at a time. So when one molecule of oxygen binds to a monomer, it basically changes the conformation. So this allows hemoglobin to have both T state and R state in a given tetramer at a time. So this model follows the concept that after binding occurs at one site in the active site, the binding affinity in the other site around the protein will increase as well. So as in this induced fit, when one molecule of oxygen will bind to one monomer, it will bring the structural change to that monomer only, but it will bring the structural change in such a way that it will increase the affinity of substrate binding or oxygen binding to the neighboring subunit. So the basic difference between these two models, the concerted model and sequential model, is that the change of the form of the subunit, like from tens state to the R state. So the basic difference is this only, that whether all the subunits will change their structure at one go, or they will change sequentially. So sequential model says that it will change sequentially, whereas the concerted model says that it will change at one time. However, neither the sequential model nor the concerted model can fully explain that how basically oxygen binds to the hemoglobin. So the properties from both the model appear in a real system. I mean, if you plot a graph and then try to analyze, neither the sequential model, which fits completely to the oxygen binding to the hemoglobin, nor the concerted model can explain completely fully that the oxygen binds to hemoglobin using this model only. So the second type of cooperativity is called negative cooperativity. So negative cooperativity can be defined like this, that when the binding of a substrate or ligand molecule inhibits the binding of other substrate or ligand molecule on either on the same subunit or on the other subunit, then it is called negative cooperativity. And the value of Hill's coefficient n is less than 1 in this case. So the best example of the negative cooperativity is phosphofructokinase. Phosphofructokinase is basically a tetrameric enzyme of glycolytic pathway, and it shows negative cooperativity. How it shows that we can understand with the example very nicely, that whenever there is excess energy in the cell, what is happening, that ATP always builds up in the cell. So this phosphofructokinase, it has two different sites for ATP binding. The one site is basically in the active site, so where it binds as a cofactor, and it has low Km. So it whenever there is low concentration of ATP, it binds there and it facilitates the reaction. However, when there is more concentration of ATP in the cell, so that means the cell have excess energy and it don't need the reaction to go on. So what it does, that ATP at high concentration, ATP bind on the allosteric site, where which has high Km value. So when at high concentration of ATP goes and bind there, and it alters the structure of active site, so that the ATP molecule cannot bind on the active site, and the substrate also cannot bind on the active site, or even if it binds very loosely, so the reaction cannot occur. In this example, which is example of negative cooperativity, you can see like this is an enzyme of glycolysis. It's a tetrameric enzyme basically, and then here how nicely the regulation occurs through allosteric control in using enzyme cooperativity. This is one of the best example of showing that. So when there is more energy in the cell, what will happen, that ATP will bind to the allosteric site, and this allosteric site is having high Km value and low affinity. So when there will be ATP concentration very high, then only it will bind to that effector site, and then what it will do, you can see in the figure in the lower panel that the the binding of ATP to the allosteric site at high concentration, it brings such a structural change that the enzyme that the substrate molecule cannot bind to the it can bind to the substrate, but the reaction cannot occur because there is a structural change, and to occur this reaction, basically ATP molecule binding of ATP molecule in the active site is required. So ATP molecule now cannot bind to the active site because of this structural change, and then enzyme substrate complex cannot form, and then reaction cannot occur. Whereas if the energy is low, so if the energy is low, it can bind to the low, it can bind as a cofactor to the active site, which is having a low Km value or at high affinity for ATP. So when the energy in the cell is very low, the cell will definitely go for glycolysis to synthesize more and more ATP, to synthesize more and more pyruvate, which can go into the mitochondria and produce more and more ATP molecule through oxidative phosphorylation also. So at low ATP concentration, it will bind to a low Km and high affinity active site in the active site, and then it brings the structural change in such a way that substrate can come and bind and form energy enzyme substrate complex, which can be shown in the upper panel, and then the reaction can occur easily. In the graph, which is plotted between the reaction velocity and fructose 6-phosphate concentration, one can see that it shows a strong very low initial lag phase, and in the sigmoidal curve at low ATP concentration one can see. However, when the ATP concentration is very high, like 1 millimolar ATP, the binding is relatively delayed, and then the reaction is also slower in that case. So students, let's now summarize what we have learned in this module. So allosteric enzyme shows cooperativity, that's what we learned, and they represent a sigmoidal curve when the curve is kinetics curve is plotted between the reaction velocity and substrate concentration. And then the important factor of cooperative enzyme, the Hill's coefficient, its value is always n is equal to 1 when then the hyperbolic curve will be plotted. So that means the enzyme is not showing any cooperativity. So but if the value of n is equal to more than 1, then it means the enzyme is showing positive cooperativity, and more the value of the positive cooperativity, the enzyme will show a better sigmoidal curve. So but if the value of n is less than 1, that means that it is showing a negative cooperativity and enzyme is not cooperative. In multimeric proteins, enzyme cooperativity itself, multimeric proteins can exhibit cooperativity in which one substrate alters the binding of other substrate, and hemoglobin is well characterized and suitable example which we read in this, which we talked about and discussed in this module, that the binding of oxygen to hemoglobin is one of the best suited, best studied model of positive cooperativity. So both types of cooperativity are involved in regulation of enzyme activity, both positive and negative cooperativity. The changes in substrate or ligand concentration exert direct change in enzymatic activity or in ligand binding by sometimes altering the structure of active sites. So binding of a small molecule can cause conformational change of varying degrees, so which is leading to even a small conformation change can have significant effects on the activity of the enzyme or protein. So molecules other than the substrate can also regulate the cooperative enzymes. For example, 2,3-bisphosphoglycerate, which is the byproduct of glycolysis produced in a shunt. It can bind to the hemoglobin and can decreases the affinity of oxygen with hemoglobin, and then facilitates the release of oxygen in tissues. So it is not neither substrate nor the inhibitor molecule, but it is some other molecule which is facilitating the release of oxygen molecule from hemoglobin. For some enzymes, increasing concentration of substrate can also decrease the enzymatic activity. It is something like feedback inhibition, so which is little bit different than feedback inhibition. So this is illustrated in case of phosphofructokinase, which we talked about, that high ATP concentration basically inhibits the reaction, and it distorts the active site structure in such a way that substrate cannot bind, and then reaction cannot occur. Thank you.